So if you look at this data, and so the first sheet, this is the first sheet I'm here, and you can see that there is different variables on top, and there's this area name, area code, type, specimen date, cumulative lab confirmed cases, cumulative lab confirmed cases rate. Now, and you can see that on the area type, if you scroll it down, you can see upper tier local authority, York, Derby, and then you can also see region, right? So these regions are different regions of England, right? And then they also use here you see nation, okay, which is England, and you can also see that there are another lower tier local authority, and you can see they're from the same date, the 16th of May to 2020. And by the way, you can see that for the entire England, there was a 37 lab confirmed cases in the 16th of May. And according to the website, what they're saying that this is not the actual increased number per day, because we still have a 3,000 a day increase. That is saying that a lot of those cases are from specimens from previous days, I think. That's my understanding. But they explicitly warn not to take these number as a sign of it has decreased to almost zero. But anyway, so you have lower tier local authorities, right? So which are more than the higher tiers. And these are from the same date. And then you can see that it also covers information from previous days, like the 15th of May and for all these, right? So now if you go to the other sheets, you can see that I have already copy and pasted using the copy and paste method of the same data into different levels. So what I mean by different levels of variability is that, first of all, you can have a regional level of analysis, that you can have all these lab confirmed cases. So now each region is just the sum of all the lower tier authorities summed together, right? But by summing them, you are basically taking out the variation between the lower tier level authority, and you sum them together to a single number. Basically, you take out all the variations. Now you can look at the variations between different regions. You can see that London has the highest number of cumulated cases at 16th of May, then Southeast, right? Then Northwest. And you can see that some regions have higher numbers, some regions have lower numbers in England. Okay, and this is the level of variation here. We're talking about regional level. Now, if you go to the next sheet, which is upper May 16th, we can see that these are the variations on cumulative lab confirmed cases between different upper tier local authority. Okay. So there are a total of how many? 150 of them in England. Okay, and see that these numbers also add up to 173,316. So is the previous region. But now you have more different kind of level of variation. You have variation of this number, which is the number of cases, across between different lower tiers of authority, upper tier local authority. Okay, and now if you move on to the next sheet, which is lower May 16, you see there are 317 lower tier authorities, and you have another level of variation. So this is what we call a hierarchical data. So region is at the highest hierarchy, level of hierarchy, and then you have lower level of hierarchy, that is upper tier and lower tier local authorities. So the same kind of statistics, which is cumulative lab confirmed cases, can have different different amount of variation at different levels. Okay, But eventually, when you move up levels, you're just basically aggregating them. Okay? But by aggregating them, you take away some of the variations, right? You're basically assuming that all the lower tiers are equally. You basically you lose the variation at the upper tiers by aggregating them to regional level. Okay. Okay. So that's what I mean by variability or variation at different levels of analysis. Okay. So I'm going back to the. Okay, so I'm going back to from the Excel file sheet to the PowerPoint. This Excel file we will be using it for the seminar, but right now let me just go back to the PowerPoint. So that's that, right? So now we're talking about how do we explore a single variable? How do we represent them? How do we depict them? So as I said earlier, if you just look at the different numbers, it doesn't make much sense. So we have to find a way to make them more, to present them in a way that we can get more information out of it. So I'm going to, we can use graphs, which is what we're going to do for this lecture. And then in the next lecture, we'll talk about how to use numbers to represent them. So here comes the idea of the concept of distribution, which is very important for further for statistical analysis. But for a single variable for a single variable on a particular sample, it is quite easy to understand. It basically says what values a variable can take and how often the variables take each values. Okay, so it can be depicted using graphs or descriptive statistics. Okay? So again, what values can it take? How often do they take each value? Okay? For a categorical variable, this is quite straightforward. So you can just take the category or label values. For example, male or female. So a variable can take either be male, female, or black, white, Asian, all these different labels, right? These labels are just labels. And it places an individual, each unit of analysis, let me put it this way, into one of the categories. And the categories should be mutually exclusive, which means that if you want in one of the group, you cannot be in the other group. So it cannot be ordered or ranked. It doesn't have any meaning. So you cannot say male is higher than female or vice versa. Doesn't make any sense. Okay, it's just categories. And depicting a categorical variable is simple, right? You can use a pie chart. For example, they did a survey for 1,200 US college students. With whom do you find it easiest to make friends? Is it opposite sex, or is it same sex, or is it no difference? So based on their responses, and this is a case where the dependent variable, right? So the behavior can be categorized according to their responses. So you have percentages, right? So you have 50% no difference, 14 is 36%. So you can say that 50% of the students think there's no difference, or more students find it easiest to make friends with the opposite sex than same sex, within those 50% who think there is a difference. So this is just a way of describing the distribution of a single categorical variable, depicting it. And you can also use bar charts. In the bar charts, you can have frequencies, right? So you have 1,200 students, and this is bar charts based on count. So you can have different counts. So this is another graphic way of depicting the distribution of a categorical variable. And you can also use percentage. Right? Now, this is getting a little bit more interesting because you lose a little bit. You lose the count by turning to percentage, but you gain a better understanding of what's going on. So now you have percentage bar charts, which is the same shape, but now the y-axis has changed into percentage from count. And you can also use a table, right? This is how you go from frequency into percentage. Basically, you have the total, and you divided the frequency by the total, and you get a percent. And then I put the term probability here, which we will address a little bit later, right? It's a percentage means that in this sample, there's 13.7% of probability, right, that someone may answer same sex, right? So that's a step forward. So you're going from empirical frequency into some kind of a probability, which is now, when you say probability, it's already moving forward towards statistical inference, or making some kind of a prediction about what's going to happen if you repeat the same study again. Okay. Now, how about a quantitative variable, right? So for the quantitative variable, it can take numerical values, and usually represent some kind of a measurement, right? It can be at least be ordered, right? At least be ordered. It can be do more, but for some variables, such as the questionnaire I did at the beginning, you can only be ordered. You can say that those who scored a 7 is higher than those who scored a 4, but you cannot say that those who scored a 7 is twice as high as those who scored 3.5. Okay, that doesn't make sense anymore. So we'll get to that the level of measurement a little bit later. But this is how you depict a quantitative variable, right? So you have all these numbers, but you can turn these numbers into what we call a histogram. So this is the numbers that we saw earlier, the days of depression before entering hospital treatment. Now, again, this is a bunch of numbers, doesn't make much sense, but if you turn it to a histogram to the right-hand side of the screen, all of a sudden it makes much more sense. Now, let's look at the histogram more carefully. So what is x-axis here? X-axis goes from 0 to 500. It is the day of the value that this variable can take, right? And this sample, it doesn't go over 500 that much, right? So, and also you can notice that these continuous, this 0.5 to 500 has been binned, so divided into small segments, right? So segments of 50. Okay, so this is also important, something called binning. Okay? And the y-axis is now the frequency. So which means that how many values within that bin has occurred in this sample. So in this case, you can tell that there's 10 cases where the days is from 50 to 100 days. Okay, for 100 to 150 days, there are more than 30 cases. So it's basically you counting the frequency of the number occurring in this data set and turning that into what we call a histogram. Now, histogram is one of the most important the concept that you need to really try to understand, because a lot of people have difficulty when they see this kind of histogram. The first thing to notice that it goes from smaller to larger. So the whole value, the values are being ordered, right? So you're actually noticing that as the day increases, the frequency decreases from 100 days on, right? The second thing is that these histograms represent frequencies. The frequencies, as we showed earlier, can also be turned into probability. If you treat the whole data set as 100%, then each column becomes a certain percentage instead of just conveying frequency information. And that is very important to keep in mind. So let me see a little bit about mechanics, then we'll take a break. So if you have grades from 15 students, right, you break the range to 10.1 intervals, right? So, and then from 40 to 100, and then you count how many grades fall into each interval. Okay, so interval is what I called bin earlier. So bin is a term that is being used by computer scientists, I think, machine learning, they call it binning. And so if you then you turn the count down into a histogram. Basically, you're mapping into these counts into a histogram. And so the height indicates the count within each bin or each interval. Okay. Notice that these intervals are exclusive by having the parentheses. So 50 will go to the second bin instead of the first bin, right? So there you have it, a histogram. A couple of important notes is that each value can only be counted once. Some information get lost because you're binning you're basically creating intervals. So you don't get to know what exactly values are in there in each interval. All you get is a count, right? Also, if you change the number of intervals, you change the look of the histogram. So for example, these are two histograms from the same data set, but you have a more fine-tuned intervals for the second one, right? So now you have a more fine-tuned looking histogram. So basically by decreasing the interval, making it smaller, and if the data allows, if the how do I say, the data is good enough, the fidelity of the data allows, then it gives you a better looking, more detailed information once you decrease the interval. Okay. Again, the final thing that I want to talk about before we go down the break is that you can turn frequency into percentage. So if you can ask yourself the question of what is the percentage of exam score equal over 90, you can count that, right? So you can draw a line here at the 90, and basically you want to know what is the size of this little bar to the right-hand side of 90 in proportion of all the areas that's being covered by the histogram. So, which is basically dividing the count of 1 by the total count, we have 6.7. So we know that the percentage of exam scores equal over 90 is 6.7%. We can ask another question, what's the percentage of exam scores up to 60? So then we draw a line at 60, and we want to count the frequencies to the left-hand side of 60, and how the percentage of that to the total number of counts. Again, we have 2 times 1 plus 1 divided by 15. We have 20%, right? So 20% of this exam scores is lower than 60, and that information can be extracted from the histogram. Now, this is a very basic example, but in the future, when it comes to statistical, more advanced, more higher level statistical procedure, you will see that this is very helpful for understanding distribution and how they're used in statistics. So let's take a 10-minute break right now. Coming back at a 3:15. Let's have make sure you have the Excel file that I sent to you earlier downloaded and open. So we're going to be using that to make a histogram in Excel. Okay. I'm going to stop the recording. I'm going to take a break. Stop recording.